You think you know the ocean. You've seen the documentaries, the whales breaching, the sharks patrolling. But wait until you hear about the creatures that evolution spent hundreds of millions of years perfecting into living weapons, shapeshifters, and immortal beings. These are not myths. These are real animals swimming in our oceans right now. Animals that punch with the force of bullets, animals that cheat death itself, animals heavier than any dinosaur that ever walked this planet. 7 species, 7 impossible abilities, all documented by science, all verified with receipts, no speculation. Just cold, hard facts pulled from peer-reviewed journals and research expeditions published between 1998 and 2025. Let's dive in. If you value documented marine biology and ocean history like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from the Schmidt Ocean Institute's 2025 colossal squid documentation filmed on March 9th near the South Sandwich Islands, published in Nature Biodiversity Data Journal on May 14, 2025, and the National Museum of New Zealand Te Papa Tongarewa's specimen archives dating back to February 2007, when they recovered that 495-kilogram colossal squid from the Ross Sea. Now back to the information. Number 1. The blue whale, the largest animal that has ever existed on planet Earth. You're standing on a basketball court, 94 feet long. Picture the entire length filled with one animal. Now add another 16 feet. That's a blue whale. 110 feet from nose to tail in the largest specimens ever measured. The record holder was caught in the Southern Ocean on March 20th, 1947. 27.6 meters long, 190 tons. That's 190,000 kilograms. The weight of 2,500 adult humans, or 30 elephants, or 15 school buses. Think about that number. 190 tons. The heaviest dinosaur we know about was probably Patagotitan mairum. 140,000 pounds, impressive. But a blue whale weighs over 400,000 pounds at peak size. Nothing on land comes close. Nothing in the fossil record matches it. The blue whale is the apex of biological mass, ever. Here's what that mass looks like up close. The heart alone weighs 180 kilograms, 400 pounds, the size of a small car. A football team could stand on the tongue without falling off. The aorta, the main blood vessel leaving that massive heart, is wide enough for a human child to crawl through. The tongue weighs as much as an elephant, 2,000 pounds of muscle. The blubber layer beneath the skin measures between 6 and 12 inches thick. This fatty tissue serves multiple purposes: insulation in freezing polar waters, energy storage during migration when the whale doesn't feed for months, buoyancy control, streamlining for efficient movement through water. The skeleton alone weighs 15 to 20 tons. The skull comprises roughly 1/4 of the total body length. The lower jaw bones, called mandibles, are massive curved structures that can measure over 5 meters long each. These bones must support the weight of 70 tons of water when the whale opens its mouth during feeding. The vertebrae, the bones of the spine, number between 55 and 88, depending on individual variation. Each vertebra in the thoracic region can weigh up to 50 kilograms. 110 pounds for a single bone. The ribs number about 15 pairs. Some extend down only partway around the chest cavity, protecting vital organs while allowing the throat to expand enormously during feeding, and they survive on the tiniest prey imaginable. Krill. Shrimp-like crustaceans barely longer than your thumb. A blue whale filters 8 tons of krill per day during feeding season, 8,000 kilograms. That's the volume of a small truck in tiny crustaceans passing through baleen plates. The whale opens its mouth, takes in enough water to fill a swimming pool, 60 to 70 tons of seawater in a single gulp, then forces it out through black baleen bristles, trapping thousands of krill inside. 70 to 395 plates line the upper jaw. Each gulp captures millions of organisms. The baleen plates themselves are made of keratin, the same protein that forms human hair and fingernails. Each plate is triangular, wide at the gum line and tapering to a frayed edge on the inner side. The frayed edges form a dense mat that acts as a sieve. Water passes through. Krill cannot. The plates are black in color, sometimes with hints of gray or brown. They grow continuously throughout the whale's life, wearing down from the constant flow of seawater and abrasive action of captured prey. A full set of baleen can weigh up to 400 kilograms when intact. The feeding process is called lunge feeding. The whale accelerates from below toward a krill concentration, dense patches that appear as dark clouds in the water. Sonar from research vessels can detect these krill swarms as distinct acoustic signatures. The whale opens its mouth at an angle between 40 and 90 degrees. The throat grooves, between 55 and 88 ventral pleats running from the chin to the navel, expand like an accordion. The throat cavity inflates to several times its resting diameter. Water rushes in. The mouth closes part way. The tongue, that 2,000-pound muscle, pushes forward against the roof of the mouth. Water is forced out between the baleen plates. Krill remains trapped inside. The whale swallows, contracts the throat pleats, returns to normal profile. The entire sequence takes between 5 and 15 seconds, depending on the depth and density of the krill swarm. Blue whales migrate thousands of kilometers every year. Summer near the poles where krill swarm in massive concentrations, winter near the equator to breed in warmer water. The journey takes months. They travel 2 to 8 kilometers per hour normally. But when threatened or chasing something, they hit 32 to 36 kilometers per hour, 22 miles per hour for a creature the length of 3 city buses. The total migration distance can exceed 10,000 kilometers one way. Antarctic blue whales travel from feeding grounds near the Antarctic ice edge to breeding grounds near the equator and back, 20,000 kilometers round trip, every year. Without stopping to feed during the northward and southward journeys, the whales navigate using several mechanisms. Magnetic field detection. The Earth's magnetic field provides directional information. Blue whales have magnetite crystals in their tissues that may function as biological compasses. Solar position. On clear days, the angle of sunlight penetrating the upper water column provides latitude information. Acoustic landmarks. Underwater topography creates characteristic sound patterns. Ocean currents generate noise. Seamounts, ridges, and trenches all produce distinct acoustic signatures. Whales learn these signatures and use them to navigate like humans use road signs. Dive depth goes down to 315 meters, confirmed by tagged specimens, 1033 feet below the surface. The theoretical aerobic dive limit, how long they can hold their breath based on oxygen in blood and muscles, is 31.2 minutes. But the longest measured dive was 15.2 minutes. They don't need longer. The feeding grounds are in the upper water column where krill concentrate. Krill perform diel vertical migration. During the day, they descend to deeper waters to avoid visual predators. At night, they rise to surface waters to feed on phytoplankton. Blue whales follow these migrations, diving deeper during daylight hours and feeding in shallower water at night. The diving physiology is remarkable. Before diving, the whale exhales, reduces buoyancy, reduces nitrogen in the lungs to minimize the risk of decompression sickness. The heart rate drops from normal cruising rate of 20 to 30 beats per minute down to 4 to 8 beats per minute during deep dives. Bradycardia. This conserves oxygen. Blood flow is redirected away from the digestive system and non-critical muscles toward the brain, heart, and swimming muscles. Myoglobin, an oxygen-binding protein in muscle tissue, is present in extremely high concentrations in marine mammals compared to terrestrial animals. Blue whale muscles are dark red, almost black, from the density of myoglobin. This protein stores oxygen and releases it slowly during dives when fresh oxygen from the lungs is unavailable. Communication travels across oceans. Blue whales are among the loudest animals on the planet. Their calls hit 188 decibels. A jet engine reaches 140 decibels. The whale's low-frequency pulses, groans, and moans can be heard by other blue whales up to 1,000 miles away in good conditions. They use sound to navigate the lightless depths. Echolocation, through millions of cubic kilometers of black water, finding feeding grounds, locating mates, coordinating movements across entire ocean basins. The vocalizations serve multiple functions. Long moans lasting 10 to 30 seconds are thought to be mating calls. Males produce these during the breeding season. The calls have fundamental frequencies between 15 and 20 Hz, well below the threshold of human hearing. Humans hear down to about 20 Hz. Blue whale calls go lower, down to 10 Hz. These infrasonic frequencies travel enormous distances because low-frequency sound waves are less absorbed by seawater than high-frequency waves. A call produced off the coast of California can theoretically be detected off the coast of Japan if ocean conditions are favorable. Pulse trains are a series of rapid low-frequency sounds. Possibly, contact calls used to maintain group cohesion. Blue whales are generally solitary or found in small groups of 2 to 4 individuals, but they coordinate over large areas using sound. Ships occasionally report seeing several blue whales converging on a single location from different directions. They're responding to calls produced by other whales, announcing a rich krill patch. Scientists use hydrophone arrays to track whale populations. Networks of underwater microphones deployed across ocean basins. These systems record whale calls continuously. Computer algorithms analyze the recordings and identify individual whales based on call characteristics. Each whale has slight variations in its call structure, analogous to a voice print. By tracking these individuals over time, researchers can estimate population sizes, migration routes, and behavioral patterns without ever seeing the whales directly. Reproduction produces the biggest babies on Earth. A blue whale calf at birth measures 20 to 23 feet long, 6 to 8 meters, weighs 6,000 to 7,900 pounds. Up to 3.5 tons, the weight of a fully grown female Asian elephant. The calf doesn't eat solid food. It drinks its mother's milk, 50 gallons per day, 225 liters of milk. Now that's 40 to 50% fat, every single day. For context, cow's milk is about 3 to 4% fat. Human milk is about 4% fat. Blue whale milk is 50% fat. This high fat content is necessary to support the calf's explosive growth rate. The milk is thick. Consistency of toothpaste. The mammary glands are located in slits on either side of the mother's genital opening. When the calf nudges the area, the mother contracts muscles around the glands and actively ejects milk. The calf doesn't suckle in the traditional sense. It positions its mouth over the gland opening and the mother squirts milk directly into the calf's mouth. This process happens underwater. The calf must surface to breathe between nursing bouts. Growth rate breaks all records. The baby gains 9 pounds per hour, 100 pounds per day, 450 kilograms per week, 1 ton every 10 days. This continues for 7 to 9 months until weaning. By that time, the calf measures 49 feet long, 15 meters. The youngster reaches sexual maturity at 5 to 15 years old. Lifespan extends beyond human generations. Scientists count layers in the whale's earplug, a waxy substance. That builds up like tree rings. Estimates suggest 80 to 90 years, maybe longer. The earplug, technically called the tympanic bulla, accumulates layers of keratin and lipids throughout the whale's life. Each layer represents roughly 6 months of growth. Light layers form during the feeding season, when the whale is consuming large amounts of food. Dark layers form during the migration and breeding season, when the whale is fasting. By counting the layers, scientists can determine the whale's age at death. The oldest blue whale on record based on earplug analysis was estimated at 110 years old. A specimen caught off Iceland in the 1930s. Recent analysis using improved techniques suggests that blue whales regularly live beyond 100 years if they survive to adulthood. The Antarctic blue whale subspecies is the largest of all. Males average 112 tons, females 130 tons. Northern Hemisphere animals are slightly smaller. Males 100 tons, females 112 tons. Eastern North Pacific males average 88.5 tons, females 100 tons. Even the pygmy blue whale subspecies, the smallest, has males weighing 83.5 to 99 tons. There are multiple, recognized subspecies. Balanoptera musculus musculus, the northern blue whale, found in the North Atlantic and North Pacific. Balanoptera musculus intermedia, the Antarctic blue whale, found in Southern Ocean waters, the largest subspecies. Balanoptera musculus brevicauda, the pygmy blue whale, found in the Indian Ocean and South Pacific. Shorter body length but still massive by any other standard. Balanoptera musculus indica, sometimes recognized as a separate subspecies in the northern Indian Ocean. Genetic studies are ongoing to clarify the relationships among these populations, and here's the brutal part. We almost lost them. Commercial whaling in the early 20th century slaughtered blue whales by the tens of thousands. The 1930 to 1931 season alone killed over 29,000 blue whales worldwide, 29,000 of the largest animals ever to exist. Shot, processed, rendered into oil. By the 1960s, blue whale populations had crashed to near extinction levels. The whaling technology of the early 20th century was devastatingly efficient. Steam-powered catcher boats, exploding harpoons tipped with grenades that detonated inside the whale's body. Factory ships that could process multiple whales per day while at sea. The blue whale's massive size, once a protection against earlier whaling, methods became a liability. Each whale represented enormous profit: oil for lamps and machinery, baleen for corsets and buggy whips, meat for animal feed, bones for fertilizer. Every part was used. The economic incentive was overwhelming. The peak of Antarctic whaling occurred during the 1930s. Norway, Britain, Japan, and the Soviet Union all operated whaling fleets in Southern Ocean waters. Between 1930 and 1940, over 350,000 blue whales were killed in the Antarctic alone. The population crashed from an estimated 250,000 individuals before industrial whaling began to perhaps low thousands by the 1960s. Some populations were reduced to a few hundred individuals. Protection came in the mid-1960s when the International Whaling Commission finally banned commercial hunting. Populations began recovering. Current estimates put worldwide numbers between 10,000 and 25,000 individuals, better than the low point, but still endangered according to the International Union for Conservation of Nature. A species that took millions of years to evolve was nearly wiped out in 50 years of industrial killing. The recovery has been slow. Blue whales reproduce slowly. Females give birth every 2 to 3 years. Calves require months of maternal care. The population doubling time, the number of years required for a population to double in size under ideal conditions, is estimated at 20 to 30 years for blue whales. Even with complete protection, recovery takes generations. Current growth rates vary by population. Some subpopulations are increasing steadily. The eastern North Pacific population grows at about 3% per year. Other populations show little to no recovery. The Antarctic population may be recovering, but precise numbers are difficult to obtain due to the vastness of the Southern Ocean and the difficulty of surveying whale populations in ice-covered waters. Recent surveys show hope. A team led by the British Antarctic Survey returned from South Georgia in 2020 with good news. They counted 55 Antarctic blue whales during their expedition. Quote, unprecedented, close quote, according to whale ecologist Jennifer Jackson. This is a place where both whaling and sealing were carried out extensively. The numbers are rising. The whales are coming back to feed where they fed a century ago before the killing started. South Georgia is located in the South Atlantic Ocean, a mountainous island roughly 170 kilometers long. It was a major whaling station. During the early 20th century, 7 shore stations processed thousands of whales per year at the peak of operations. The ruins of these stations still stand. Rusting boilers, collapsed buildings, massive iron cauldrons where whale blubber was rendered into oil. The bones of thousands of whales still litter the beaches around the old stations, reminders of the industrial slaughter that occurred here. But here's the reveal that reframes everything. In 2024, researchers Motani and Pyanson published calculations showing that a blue whale at maximum size doesn't just weigh 190 tons. Based on volumetric analysis and regression, Data from hundreds of measured specimens, the true maximum could reach 199 to 200 tons, 200,000 kilograms. The animal pushes the absolute physical limits of what living tissue can achieve on Earth. Blood pressure that would burst smaller animals' hearts. Bone density and muscle fibers engineered by evolution to support a creature that makes every land animal look fragile. The blood pressure in a blue whale's circulatory system must overcome enormous gravitational forces. When a whale is oriented vertically, diving or surfacing, the pressure differential between the head and tail is substantial. The heart generates pressure around 150 to 200 millimeters of mercury, higher than human blood pressure, but not proportionally higher considering the whale's size. The secret is the architecture of the circulatory system. Large diameter blood vessels reduce resistance. Muscular valves in the veins prevent backflow. Specialized structures called retia mirabilia, networks of intertwined arteries and veins, help regulate temperature and pressure in different body regions. The heart itself is a four-chambered structure, like all mammalian hearts. 2 atria receive blood from the body and lungs. 2 ventricles pump blood to the lungs and body. The left ventricle, which pumps oxygenated blood to the body, has walls several inches thick, composed of dense cardiac muscle capable of generating enormous pressure with each contraction. The heart beats slowly, 20 to 30 beats per minute during normal activity. Each beat moves hundreds of liters of blood through the system. The cardiac output, the volume of blood pumped per minute, can reach 500 liters per minute during strenuous activity. That's enough to fill a standard bathtub every minute. You want to see one? Dana Point, California. The highest-rated whale-watching company there is Dana Wharf Whale Watching. Daily tours, experienced captains, naturalists on board. They guarantee you see a marine mammal or you come back free. The blue whales migrate through those waters. You'll stand on a boat looking at an animal that is functionally infinite compared to human scale. An animal that breathes air like you but weighs as much as a small building. An animal that feeds on creatures so tiny you could hold dozens in one hand, but consumes metric tons of them per day. The largest animal that has ever lived, swimming in the Pacific Ocean, right now. The California coast is part of the eastern North Pacific blue whale population's migration route. Whales travel north from breeding areas off Mexico and Central America to feeding grounds off California, Oregon, Washington, and British Columbia during summer months. The continental shelf waters along California are rich in krill and other zooplankton due to upwelling. Cold, nutrient-rich water rises from depth, fertilizing surface waters and triggering phytoplankton blooms. Krill feed on the phytoplankton. Blue whales feed on the krill. The timing of the whale migration tracks the peak krill abundance. Dana Point specifically is located where deep submarine canyons approach the coast. These canyons funnel nutrient-rich water toward shore and concentrate prey species. Marine mammals, including blue whales, gray whales, humpback whales, dolphins, and seals, congregate in these productive waters. Whale watching success rates are high. Over 90% of tours encounter at least one species of whale or dolphin. The blue whales are most common from May through November. They feed actively in the channel between the mainland and the Channel Islands, sometimes approaching within a few hundred meters of the whale-watching boats. Number 2. The colossal squid, the creature with the largest eyes in the animal kingdom. March 9th, 2025. South. Sandwich Islands in the South Atlantic Ocean. Research vessel Falkor from the Schmidt Ocean Institute, 600 meters below the surface. 1,968 feet of black water pressing down. The remotely operated vehicle, Sebastian, sweeps its lights across the deep. And there, floating, transparent, 30 centimeters long, 1 foot, a juvenile colossal squid, Mesonychoteuthis hamiltoni. This is the first confirmed footage of a colossal squid alive in its natural habitat. First time since the species was formally identified and named 100 years ago. This year marks the centennial. And on March 9th, expedition chief scientist Michelle Taylor and her team finally captured what scientists have been hunting for since 1925. The South Sandwich Islands are a volcanic arc in the South Atlantic. Remote, uninhabited, perpetually cold, surrounded by some of the roughest seas on the planet, the Scotia Sea, where the Antarctic circumpolar current sweeps eastward, unimpeded by land masses. Storms form quickly. Waves reach 15 meters during winter. Research vessels rarely venture here. But the Schmidt Ocean Institute's Falkor is equipped for extreme conditions: reinforced hull, dynamic positioning thrusters that maintain station against current and wind, and Sebastian, a remotely operated vehicle rated to 4,500 meters depth. The ROV descended on a routine deep-sea transect, cameras recording, sonar mapping the bottom topography. Scientists aboard the ship watched monitors showing the live feed from 600 meters below. The water was dark, occasional glimpses of gelatinous creatures drifting past the lights. Then Sebastian's camera captured something different, larger than the typical jellies, more structured, swimming with purpose, the juvenile colossal squid. Let's establish what we know. The colossal squid is not the longest invertebrate. That title goes to the giant squid, Architeuthis dux, which can reach 5 meters for females, males slightly shorter with feeding tentacles adding another 10 meters. Total maximum length around 16 feet. But the giant squid is slender, built for speed. The colossal squid is built for mass. Maximum confirmed length for a colossal squid is 4.2 meters, 14 feet. Maximum confirmed weight is 495 kilograms, 1,091 pounds. The heaviest cephalopod ever recorded. This specimen was caught in the Ross Sea in February 2007. Female. Initially estimated at 450 kilograms before complete thawing. After 14 months frozen, the specimen was thawed in a saltwater bath. At the Museum of New Zealand, Te Papa Tongarewa. Final measurements: 495 kilograms, mantle length 2.5 meters, total length only 4.2 meters. Short, because the tentacles and arms shrank post-mortem from dehydration. In life, this animal was probably longer. The thawing process took place in a custom-built tank over the course of several days. The specimen had been frozen solid at -20 degrees Celsius for over a year to preserve it, until proper facilities were available for examination. Marine biologists, technicians, and media representatives watched as the ice slowly melted away. The thawed specimen was examined in detail. Every measurement recorded. Tissue samples taken for genetic analysis. The beak removed and measured. The eyes carefully examined and photographed. This was only the second complete adult colossal squid ever examined by scientists. The first was a smaller male caught in 2003, also in the Ross Sea. But even that 495-kilogram monster is not the biggest. The beak, the hard chitinous mouth part that doesn't decompose, tells us more. Scientists find colossal squid beaks in sperm whale stomachs. Sperm whales hunt colossal squid in the deep. The beaks survive digestion, and some of those beaks found in whale stomachs are considerably larger than the beak from that 495-kilogram specimen. Estimates based on beak size suggest colossal squid could reach 600 to 700 kilograms, 1,300 to 1,500 pounds, maybe heavier. We just haven't found one yet. The beak measurements follow allometric scaling. The relationship between beak size and total body mass is well understood from smaller squid species. By measuring the beak dimensions, particularly the lower rostral length and the hood length, scientists can estimate the body mass of the squid that produced the beak. The largest colossal squid beaks recovered from sperm whale stomachs measure significantly larger than the beak from the 495-kilogram female. Extrapolating from the scaling relationship suggests individual colossal squid exceeding 700 kilograms exist in the Southern Ocean. These animals have never been caught or photographed, but the beaks prove they're out there. The eyes. This is where the colossal squid wins the record that nothing else can touch. The eyes on the February 2007 Ross Sea specimen measured 27 to 30 centimeters in diameter when alive, 11 to 12 inches. Dinner plate sized. The partially collapsed specimen after death showed evidence the eyes were likely 30 to 40 centimeters across when the animal was swimming. 16 inches in diameter. The pupil alone measured 9 centimeters, 3.5 inches. The eye structure is complex. A large spherical lens focuses light onto the retina. The retina contains photoreceptor cells that convert light into neural signals. Unlike mammalian eyes with a blind spot where the optic nerve exits, squid eyes have the optic nerve fibers arranged, so there is no blind spot. The entire retinal surface is functional. This gives better resolution and coverage. The squid sees clearly across a wider field than a vertebrate, with equivalent eye size. The sclera, the tough outer layer of the eye, must be thick enough to maintain the eye's shape under pressure. At the depths where colossal squid live, pressure can exceed 60 atmospheres. 600 meters equals about 60 times atmospheric pressure at sea level. The sclera of the colossal squid's eye is reinforced with cartilaginous rings. These rings prevent the eyeball from collapsing. They also provide attachment points for the muscles that move the eye within the socket. Why eyes that enormous? The deep ocean is not completely dark. Bioluminescence. Many deep-sea creatures produce light through chemical reactions in specialized organs. Prey glows. Predators glow. And if you're a colossal squid, your biggest threat is sperm whales. Whales are huge. They displace massive volumes of water. And in the deep, that displacement creates bioluminescent trails as the whale's body disturbs tiny glowing organisms. A colossal squid with dinner plate eyes can detect that faint glow from far away, spot the whale coming, and escape before the whale closes the distance. The giant eyes serve another function, detecting the silhouettes of prey against the faint downwelling light from the surface. Even at 600 to 1,000 meters depth, some light penetrates from above. Not enough for human eyes to detect, but for an eye 30 centimeters in diameter, with a massive lens that can gather every available photon, that faint light is sufficient. Prey swimming above the squid blocks that light creates a dark shadow. The squid sees the shadow, tracks it, attacks from below. The colossal squid's range extends thousands of kilometers north of Antarctica to southern South America, southern South Africa, and the southern tip of New Zealand. Primarily an inhabitant of the circumantarctic Southern Ocean, sightings cluster near the Cooperation Sea and Ross Sea because of prey and competition. Antarctic toothfish. A study between 2011 and 2014 examined 8,000 Antarctic toothfish brought aboard trawlers. 71 showed clear signs of attack by colossal squid, bite marks, sucker scars. The squid hunts them, but sometimes the toothfish wins. Antarctic toothfish, Disosticus morsoni, are large predatory fish that can reach 2 meters in length and 100 kilograms in weight. They inhabit the same depth range as colossal squid. Both species compete for similar prey, and they prey on each other. The toothfish have large teeth and powerful jaws. When a colossal squid attacks, the toothfish defends itself, sometimes successfully. The scars on recovered toothfish show sucker marks arranged in distinctive patterns. Each sucker leaves a circular scar. The pattern matches the arm arrangement of colossal squid. Some toothfish have scars indicating multiple attacks over time. They survived previous encounters and healed. Vertical distribution correlates with age. Young squid between 0 and 500 meters. Adolescents 500 to 2,000 meters. Adults live primarily in the mesopelagic and bathypelagic regions. The open oceans, middle and deep zones, down where pressure is crushing and food is scarce. The life cycle likely follows a typical deep-sea squid pattern. Eggs are laid in deep water. They sink to the bottom or remain neutrally buoyant at depth. Hatching produces paralarvae, tiny squid that immediately begin feeding on microscopic prey. As they grow, they move between depth zones following food resources. Juveniles are transparent or semi-transparent to avoid visual predators. As they mature, they develop pigmentation and descend to deeper water where adult prey is more abundant. Reproductive maturity is reached at some undetermined age. Mating occurs in deep water. The life cycle completes. Total lifespan is unknown. Estimates based on related species suggest colossal squid may live several years, possibly up to 10 years or more. Diet consists mainly of small fish, lanternfish, deep-sea smelt. Stomach contents confirm this, but they also attack larger fish, those toothfish attacks, and there's evidence of cannibalism. A study in Prids Bay found squid remains inside a female colossal squid's stomach. They'll eat their own kind. Lanternfish family Myctophidae are among the most abundant vertebrates on Earth by biomass. They perform diel vertical migration, spending daylight hours at depth and migrating to surface waters at night to feed. Colossal squid intercept them during these migrations. The squid's large eyes detect the bioluminescent photophores that lanternfish use for communication and camouflage. Once detected, the squid extends its feeding tentacles, grabs the fish, pulls it back to the arm crown. The arms manipulate the prey toward the beak. The hunt is efficient, quick. The squid can catch multiple fish in a single feeding bout. Predators include sperm whales and sleeper sharks, but the colossal squid fights back. Sperm whale bodies sometimes show circular scars from the squid's suckers, sucker rings lined, with sharp rotating hooks. The squid doesn't go quietly. It latches on, it tears flesh. The whale usually wins because the whale is bigger and has teeth designed for cephalopods, but the squid makes the whale work for it. Sperm whales, Physeter macrocephalus, are the largest toothed predators on Earth. Males can reach 18 meters in length and weigh 50 tons. They dive to depths exceeding 2,000 meters, hold their breath for over 90 minutes, hunt squid in the deep. Their primary prey is giant squid and colossal squid. The whales have conical teeth in the lower jaw. The upper jaw has sockets that the lower teeth fit into. This dental arrangement is designed for gripping slippery prey. Once the whale clamps down, the squid cannot escape. But before that happens, the squid fights. The hooks on the colossal squid's arms and tentacles are sharp, curved. They rotate on a ball joint. When they sink into whale flesh, they swivel to lock in place. The whale must tear the arm off to remove the hooks. Many sperm whales bear circular scars from these hooks. Some scars are decades old. The whales survive, heal, continue hunting. But the scars are permanent reminders that colossal squid are dangerous prey. Sleeper sharks, Somniosus species, are large, slow-moving sharks that inhabit cold, deep water. They can reach 7 meters in length. They scavenge and hunt. Colossal squid are within their prey range. But there is little direct evidence of sleeper sharks eating colossal squid. No squid remains have been found in sleeper shark stomach content studies from Antarctic waters. This might mean sleeper sharks rarely encounter colossal squid, or it might mean the squid are too fast or too well defended for sleeper sharks to catch them regularly. The hooks are key. Colossal squid have swiveling hooks on the middle of their 8 arms. Each hook rotates independently. When the squid grabs prey, the hooks dig in and swivel to lock the grip. No escape. The 2 longer tentacles also have hooks at the clubs on the end. The squid shoots the tentacles out to snatch prey up to 10 meters away, 33 feet, then drags the prey to the arms. The arms pass it to the beak. The beak, sharp as a parrot's and made of chitin, slices the prey into chunks. Inside the beak is the radula, a tongue with rows of teeth. It grinds the chunks smaller before they pass into the throat. The beak is composed of chitin, a polymer of glucosamine units, extremely hard, resistant to degradation. The beak has 2 parts, upper and lower. The lower beak is larger and fits inside the upper. The sharp edges come together like scissors. The squid can generate enormous bite force, enough to slice through fish bones and tough tissue. The beak is surrounded by a muscular mass called the buccal mass. This structure manipulates prey and positions it for cutting. Once the prey is reduced to small pieces, the radula takes over. The radula is a ribbon-like organ covered with tiny teeth arranged in rows. It moves back and forth like a rasp, grinding food into a paste. The paste is mixed with digestive enzymes from the salivary glands. Partial digestion begins in the mouth. The partially digested food passes down the esophagus, but in squid, the esophagus passes through the center of the brain. This means the squid must swallow small pieces. Anything too large could damage the brain. The stomach receives the food. Digestion continues, nutrients are absorbed. Waste is expelled through the anus near the siphon. Back to that March 9, 2025 footage. The juvenile filmed by Sebastian was transparent. Glass squid family, Cranchidae. As juveniles, colossal squid are nearly see-through. Only chromatophores, color-changing cells interrupt the transparency. Scientists watching the footage saw those chromatophores. That means the squid can switch between transparent and opaque, probably has fine control over which body regions change. Camouflage even when you're already invisible. The footage also revealed something scientists suspected, but hadn't confirmed until now. This was a teenage squid, not a baby, not an adult. Mid-developmental stage based on physical features. Proportions intermediate between juvenile and mature forms. Aaron Evans, independent scientist and glass squid expert, said, quote, "This is not the giant animal we'd expect the colossal squid to be. It's not a baby either," close quote. Published in National Geographic on September 5, 2025. The proportions visible in the footage show a mantle length of approximately 15 to 20 centimeters. The arms and tentacles are relatively short compared to the mantle. In adult colossal squid, the arms and tentacles are proportionally longer. The eyes are large, but not yet the massive organs seen in adults. The skin shows developing pigmentation patterns. Dark patches beginning to form on the mantle. This indicates the squid is transitioning from the transparent juvenile phase to the pigmented adult phase. Based on comparisons with other squid species, this individual is probably 1 to 2 years old, halfway through its expected lifespan. Here's the brutal reality. We still don't know much. Only a handful of complete colossal squid specimens exist. Fewer than 30 documented whole specimens. Hundreds of beaks from whale stomachs. Fragmentary remains caught in trawl nets. Until March 9, 2025, nobody had filmed one alive in the deep. We know more about the surface of Mars than we know about the colossal squid's behavior, life cycle, mating, and hunting strategies. The deep ocean keeps its secrets, but we know one thing for certain. Somewhere in the black water below Antarctica, colossal squid larger than any we've measured are swimming right now. 600-kilogram animals, maybe 700, with eyes the size of basketballs, watching for the faint bioluminescent glow of approaching sperm whales. The largest invertebrates on Earth, and we only just filmed one for the first time, 100 years after naming the species. The ocean is vast, the darkness is absolute, and the colossal squid lives where humans can barely reach. Number 3, the immortal jellyfish, the only animal that can reverse its own aging. Every living thing dies, cells age, DNA degrades, organs fail. The arrow of time points one direction. Birth, growth, reproduction, death, except for one animal, Turritopsis dohrnii, the immortal jellyfish. It bypasses death. It rewinds its life cycle. It becomes young again. This is not speculation. This is documented cellular biology observed under microscopes and confirmed in peer-reviewed journals since the 1990s. Let's walk through exactly what happens. Turritopsis dohrnii measures 4.5 millimeters wide and tall, smaller than your pinky fingernail. It lives in temperate to tropic waters worldwide. Mediterranean, Caribbean, Pacific, Atlantic, Japan, Panama, Brazil, Gulf of Mexico, Turkey, California, everywhere. Because it hitchhikes on ships and survives long trips in ballast water better than almost any other organism. The jellyfish has a standard hydrozoan life cycle, starts as a planula, a free-swimming microscopic larva. The planula settles on the sea floor, transforms into a polyp, a stationary tube-shaped organism attached to rock or substrate. The polyp is asexual. It buds off more polyps, builds a colony. When the colony reaches certain size, it produces medusae, the swimming bell-shaped form we recognize as jellyfish. The medusae are sexual, male and female. They release sperm and eggs into the water. Fertilized eggs become planula. The cycle continues. But Turritopsis dohrnii has one more stage. When the medusa is stressed, damaged, starving, or old, it doesn't die. It settles on the sea floor, reabsorbs its tentacles, loses the ability to swim, becomes a blob, a cyst. Over the next 24 to 72 hours, this cyst transforms back into a polyp. A juvenile polyp identical to the one that originally budded the medusa. The medusa just rewound its development. This process is called transdifferentiation. Adult cells, specialized for specific tissues, become completely different types of specialized cells. Muscle becomes nerve. Nerve becomes digestive. Digestive becomes reproductive. The entire body reorganizes at the cellular level. It's not healing. It's not repair. It's metamorphosis in reverse. The discovery happened by accident. 1980s. Students Christian Summer and Giorgio Bavistrello in Italy. They collected Turritopsis polyps, kept them in jars, monitored them until medusae were released, expected the medusae to mature, spawn, die, standard life cycle. When they checked the jars later, they found newly settled polyps, young polyps, no fertilization, no larval stage. The medusae had transformed back. They continued observing. When stressed, medusae would fall to the bottom of the jar and transform into polyps without any sexual reproduction. The medusa just opted out of dying. The discovery, aided by the nickname immortal jellyfish, captured global attention once the findings were properly published and replicated. Follow-up studies confirmed it. Stefano Pirino jumped into the research, planned experiments tracking the transdifferentiation at cellular level. Published in 1996 in the Bulletin of Marine Science, the paper documented the complete reversal process, included photographs showing the stages of transformation. Within the scientific community, the paper generated immediate interest. Here was an animal performing a biological feat that contradicted fundamental assumptions about development and aging. Development was supposed to be unidirectional. Once a cell differentiated, it remained differentiated. Turritopsis violated that rule. Then Japanese scientist Shin Kubota started rearing them in his lab at Kyoto University. Only one scientist has managed to keep Turritopsis thriving long-term, Kubota, since the 1990s. During a 2-year period, his colony rebirthed itself 11 times, 11 complete life cycle reversals. Medusa to cyst to polyp to colony to medusa. Again and again. Kubota's laboratory setup is meticulous. He maintains multiple tanks at precise temperatures, feeds the medusa live Artemia nauplii, baby brine shrimp, daily, changes the water frequently to maintain quality, monitors salinity, pH, and oxygen levels continuously. The jellyfish are delicate. Any fluctuation in conditions can stress them and trigger premature transformation or death. Kubota has refined his methods over decades. He can reliably induce transformation by manipulating environmental conditions, temperature shock, salinity change, starvation. Each method triggers the reversal process. Kubota regularly appears on Japanese television talking about his immortal jellyfish. He's recorded several songs about them, sings them at the end of his conference presentations. The man loves his jellyfish. He sees them as holding the key to understanding aging and potentially developing interventions for human age-related diseases. He's given hundreds of presentations worldwide, written papers, collaborated with researchers in genetics, molecular biology, and bioinformatics. His enthusiasm is infectious, and his dedication has made him the world's foremost expert on Turritopsis dohrnii. The cellular mechanism is extraordinary. A 2022 study reported the key molecular mechanisms. Researchers compared the newly presented genome of Turritopsis dohrnii with a similar, but non-rejuvenating jellyfish, found genes involved in DNA replication and repair, stem cell renewal, chromatin remodeling and organization, matrix metalloproteinases, embryonic development pathways, all upregulated during reverse development. The genome sequencing revealed Turritopsis has multiple copies of genes involved in DNA repair, more copies than related jellyfish that cannot perform life cycle reversal. These genes encode proteins that fix damaged DNA, replace mutated sequences, maintain telomere length. During transformation, the expression of these genes increases dramatically. The jellyfish is essentially repairing all accumulated cellular damage and resetting its cells to a younger state. When the medusa transforms back, gene expression shifts dramatically. Genes involved in membrane transport, nervous system, components of the mesoglea, and muscle contraction get suppressed. Genes involved in chitin metabolism and formation of primary germ layers get activated. The colonial polyp and reversed polyp show significant differences. The reversed polyp is enriched with genes for chromatin remodeling. Embryonic development. The organism literally resets its developmental clock. Chromatin remodeling involves changing how DNA is packaged inside the nucleus. DNA wraps around histone proteins to form nucleosomes. These nucleosomes can be tightly packed, making genes inaccessible, or loosely packed, allowing genes to be expressed. During transdifferentiation, Turritopsis modifies the histone proteins through acetylation and methylation. These chemical modifications change the chromatin structure and activate genes that were silenced in the adult stage. Embryonic genes turn back on. Adult-specific genes turn off. The cell reverts to a more pluripotent state. Telomeres, the protective caps on the ends of chromosomes that shorten with each cell division. Telomeres shorten as you age. Turritopsis maintains telomere length through specific cellular processes during life cycle reversal, effectively resetting cellular aging. Published in PMC Journal on October 23, 2021. The species possesses unique mechanisms related to telomere maintenance that play a significant role in its regenerative abilities. Telomeres are repetitive DNA sequences at the ends of chromosomes. They protect the coding regions of the chromosome from degradation. Each time a cell divides, telomeres shorten slightly. After many divisions, telomeres become so short that the cell can no longer divide. It enters senescence or dies. This is the Hayflick limit, the maximum number of times a cell can divide. In most animals, telomere shortening contributes to aging, but Turritopsis has high levels of telomerase, an enzyme that adds DNA sequences back onto telomeres. During transformation, telomerase activity spikes. Telomeres are rebuilt. The cells regain the ability to divide many more times. Diet. They eat zooplankton, fish eggs, larvae, small mollusks, use tentacles lined with nematocysts, stinging cells, inject venom, paralyze prey, pull it to the mouth. They can also expand their bells to create negative pressure, sucks in water and prey, creates thrust for movement, up, down, sideways, efficient hunters for their size. The nematocysts are complex organelles unique to cnidarians. Each nematocyst consists of a coiled thread inside a capsule. A trigger hair, the nidocyl, extends from the capsule. When prey touches the nidocyl, the capsule rapidly fires. The thread everts in less than a millisecond, one of the fastest cellular processes in nature. The thread penetrates the prey's tissue, delivers venom. The venom contains neurotoxins that paralyze the prey and enzymes that begin digestion. The jellyfish's tentacles contract, pulling the immobilized prey toward the mouth. Predators eat them: penguins, fish, sea turtles, sea anemones, other jellyfish. The immortal jellyfish can escape aging, but it can't escape being eaten. Most individuals in the wild die from predation or disease long before they get the chance to reverse their life cycle. Immortality is potential, not guaranteed. In the wild, Turritopsis faces constant threats. Fish that specialize in eating gelatinous prey cruise through jellyfish aggregations, consuming hundreds per day. Sea turtles, especially leatherbacks, eat jellyfish almost exclusively. They have papillae in their throats that point backward, preventing jellyfish from escaping once swallowed. Sea anemones capture drifting jellyfish with their tentacles. Other jellyfish species prey on Turritopsis. Even though Turritopsis has stinging cells, larger predatory jellyfish have stronger venom and more tentacles. The odds are against any individual jellyfish surviving long enough to need its immortality mechanism. Geographic spread is remarkable. Originally described from the Mediterranean in 1883 as Dendroclava dorni by Weismann, later reclassified as Turritopsis. Genetic studies by Milieta and Lesios in 2009 found Turritopsis dorni in wildly separated locations: Indian River Lagoon, Florida, Caribbean coast of Panama, Pacific coast of Panama, Japan. High degree of genetic similarity among populations. disjunct distribution that couldn't be explained by natural dispersal, human-mediated introduction via ships, ballast water transport, hull attachment, silent invasion. Ships take on ballast water in one port and discharge it in another to maintain stability. This ballast water contains plankton, algae, bacteria, and small invertebrates. Turritopsis polyps can attach to surfaces inside ballast tanks, or planular larvae can be sucked up with the water. When the ship reaches its destination and discharges ballast, the jellyfish are released into a new environment. If conditions are suitable, they establish a population. Over decades and centuries, Turritopsis has spread globally through shipping traffic. It's now found on every continent except Antarctica. The jellyfish adapts to local conditions. Tropical regions like Panama, 8 tentacles. Temperate waters like Mediterranean and Japan, 24 or more tentacles. Not clear why they differ. Possibly temperature-related, possibly food availability. Research ongoing. Temperature affects metabolic rate. Warmer water speeds up metabolism. Jellyfishes in warm water may produce fewer tentacles because they can catch enough prey with less surface area. Colder water slows metabolism. More tentacles provide more hunting surface, captures more of the less abundant prey. This is one hypothesis. Another is that different populations represent cryptic species, genetically similar but not identical. The morphological differences reflect genetic divergence that hasn't been detected yet. Genetic sequencing at higher resolution might reveal multiple species currently classified as Turritopsis dohrnii. Invasive species often cause problems. Zebra mussels in North America, cane toads in Australia, hippos in Colombia. But Turritopsis dohrnii doesn't have perceivable negative impact. It's tiny, translucent, feeds on plankton. Doesn't outcompete native species, doesn't damage ecosystems, just quietly spreads around the world, living, reversing, living again. The genome is being investigated. Decoding it will be the first step toward finding the immortality switch, the molecular mechanism that allows a resetting of developmental information across all cells, leading to ontogeny reversal. Medical implications are obvious. If we understand how Turritopsis rewires cells, we might develop treatments for age-related diseases, regenerate damaged tissue, replace cells destroyed by disease or injury, stem cell research, transdifferentiation research, all informed by a jellyfish smaller than your fingernail. The medical applications are profound. Imagine treating heart disease by converting scar tissue back into healthy heart muscle, or reversing neurodegenerative diseases by regenerating brain cells, or curing blindness by rebuilding damaged retinas. These are not science fiction scenarios. They're logical extensions of understanding cellular reprogramming. Turritopsis does all of this naturally. It converts one cell type into another, repairs damage, resets aging markers. If we can identify the genetic switches and molecular pathways that enable this, we can potentially replicate them in humans. The research is in early stages, but the potential is enormous. But here's the twist. It's not easy to work with. All research relies on material collected in the field. You can't mass culture them. Laboratory animals need to be easy to rear. Turritopsis is not easy, sensitive, requires perfect conditions, daily inspection to ensure they've properly digested their food. Artemia cysts, brine shrimp eggs. Most scientists can't keep them alive for extended periods. Only Kubota has cracked the code. The difficulty in culturing Turritopsis limits research progress. Without stable laboratory populations, experiments are difficult to replicate. Sample sizes remain small. Genetic studies require many individuals for statistical power. Without them, conclusions are tentative. Researchers worldwide are trying to develop better culturing techniques, temperature control systems, automated feeding mechanisms, water quality monitoring. Progress is slow, but necessary if we want to fully exploit Turritopsis as a model organism for aging research. And here's what stops it from being truly immortal. The reversal must be triggered. Stress, damage, starvation, physical threat. If none of those happen, the medusa just continues its normal life cycle. Reproduces, eventually dies from accumulated cellular damage or predation. The reversal is an emergency response, not an automatic process, and every time the Medusa reverses, it's genetically identical to the previous version. Same DNA, same individual. It's not reincarnation, it's regeneration. Think about it. One individual Teratopsis dohanii could theoretically persist for hundreds or thousands of years if conditions allow repeated reversals and it avoids predators. Same genetic individual, cycling through stages, polyp to Medusa to cyst to polyp, endless, barring accidents. That's as close to immortality as biology gets. The implications are staggering. A single jellyfish could outlive civilizations, persist through climate changes, survive mass extinctions. As long as it continues reversing when threatened and avoids being eaten, it never dies of old age. In theory, a Teratopsis born in the Mediterranean in Roman times could still be alive today, having reversed hundreds of times. The same individual DNA, the same jellyfish, persisting through 2,000 years of history. We have no way of knowing, no way of tracking individual jellyfish over such time spans, but it's biologically possible. The discovery is barely 30 years old, 1996 first publication. We're still figuring out the mechanisms, but the fact is undeniable. Teratopsis dohanii can reverse its aging, become young again, repeat indefinitely. The only animal on Earth that bypasses the one-way arrow of development. Verified, documented, immortal. If you value documented marine biology and ocean history, like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from the National Institutes of Health (PMC) databases, October 2021 publication titled Cellular Reprogramming and Immortality Expression. Profiling reveals putative genes involved in Teratopsis dohanii's life cycle reversal, and the American Museum of Natural History's documentation of transdifferentiation mechanisms updated in 2022 after the genome sequencing study by Hasegawa's team comparing Teratopsis dohanii with non-rejuvenating glass squid species. Now back to the information. Number 4. The mantis. Shrimp, the animal with the fastest punch in the ocean. Imagine punching a wall 2,000 times at the speed of a bullet, and your fist never breaks. That's the mantis shrimp, 4 inches long, bright colors, lives in coral reefs and sandy bottoms across tropical oceans, and packs one of the most devastating weapons in the animal kingdom. The peacock mantis shrimp, Odontodactylus ciliarus, native to the Pacific and Indian Oceans, 18 centimeters long maximum, 7 inches, weighs around 0.6 kilograms, 1 pound 5 ounces, about as heavy as a basketball. Shell is a kaleidoscope of green, orange, blue and red, leopard-like spots on the legs. Beautiful animal, absolutely gorgeous, and functionally a living hammer. The punch, the club, the dactyl, the hammer-like appendage on the mantis shrimp's raptorial arms. When the mantis shrimp strikes, the club edge travels at about 50 miles per hour, 80 kilometers per hour, under 0.003 of a second to complete the strike, 23 meters per second documented, 75 feet per second, same acceleration as a .22 caliber bullet, force of 1,500 newtons, over 2,500 times the shrimp's weight. Let that sink in. 2,500 times body weight. If a human could punch with proportional force, we'd generate strikes exceeding 150,000 pounds of force. Enough to demolish concrete walls, enough to punch through steel plate. The mantis shrimp does this routinely, every time it hunts, every time it defends its burrow, every time it fights a rival. The club hits, the prey dies, or the shell cracks, or the rival retreats. The strike is decisive. Dr. Sheila Patek at Duke University studied this, started filming peacock mantis shrimp at UC Berkeley, needed high-speed cameras. The systems she was using weren't fast enough to capture the strike accurately. BBC crew filming for the series Animal Camera offered to lend her a super high-speed camera. Patek captured footage slowed down over 800 times. Published in 2004 in Nature, what she found was staggering. The strike is one of the fastest limb movements in the animal kingdom, and it happens underwater. Water is much denser than air, 800 times denser. The drag is enormous. Any martial artist would struggle to punch quickly in water. The mantis shrimp finishes its strike in under 0.003 of a second, outpunching land animals in a medium that should slow it down. The mechanism: power amplification. The mantis shrimp doesn't generate that speed with muscle alone. Impossible. Muscles can't contract that fast. Maximum contraction velocity for muscle tissue is limited by the sliding filament mechanism. Actin and myosin proteins slide past each other, shortening the muscle fiber. This process has a speed ceiling. The mantis shrimp exceeds that ceiling by an order of magnitude. How? By using a latch and spring system. The mirus segment of the raptorial appendage contains muscles that cock the arm. Pull it back, lock it in place with a latch. A small saddle-shaped structure in the arm compresses like a spring, stores energy. When the latch releases, the spring expands, provides extra push, accelerates the club at up to 10,000 times the force of gravity. 10,000 Gs. Fighter pilots black out at 9 Gs. The mantis shrimp's club experiences 10,000 and stays intact. The saddle shape is key. Saddle-shaped springs are well known to engineers and architects, unusual in biological systems. But the Venus flytrap uses a similar mechanism, stores energy in curved structures, releases it explosively. Published in Proceedings of the National Academy of Sciences, August 17, 2021, researchers from Harvard University and Duke University built a robot mimicking the mantis shrimp's latch mechanism. 1.5 grams, shrimp scale. The robot clocked in at 26 meters per second in air, acceleration equivalent to a car reaching 58 miles per hour in 4 milliseconds. In water, about 5 meters per second, quarter of the mantis shrimp's speed. Nature still wins. The robot experiments revealed the efficiency of the biological system. The mantis shrimp's spring mechanism captures nearly 100% of the energy from muscle contraction, stores it without loss, releases it instantaneously. Human-engineered springs lose energy to heat and friction. The mantis shrimp's spring is nearly perfect. The materials composing the spring have unique properties: stiffness combined with flexibility, resistance to fatigue, the ability to compress and expand millions of times without degrading. Materials scientists are studying these properties to develop new composites for industrial applications. But the strike isn't just about the club hitting the prey. There's a second hit: cavitation. The appendage moves so fast through water, it creates an area of low pressure. A bubble forms. When the bubble implodes, it releases incredible amounts of energy: light, sound, heat. The cavitation bubble's collapse hits with almost the same force as the club itself. A single cavitation bubble is proportional to the firing of a .22 caliber bullet. 1-2 punch, physical strike, cavitation strike, both within milliseconds. This was Patek's second major discovery: small flashes of light upon impact, cavitation bubbles visible on high-speed footage. The phenomenon occurs when an object moves through water fast enough to create extreme velocity gradients. The pressure behind the club is atmospheric. The pressure immediately in front drops, below the vapor pressure of water. Water vaporizes, forms a bubble. The bubble collapses almost instantly. The collapse generates temperatures exceeding 5,000 degrees Celsius, hotter than the surface of the sun, for a nanosecond, produces a flash of light called sonoluminescence, and releases a shockwave that propagates through the water. The Spira species of mantis shrimp, with long pointed appendages for stabbing fish, don't create cavitation bubbles. Their appendages are too sleek. But smasher species with blunt clubs do. The geometry and speed create perfect conditions for bubble formation. The prey doesn't stand a chance. Mollusks, crabs, shrimp, other mantis shrimp. The club smashes through shells, cracks them open. The mantis shrimp feeds on the soft tissue inside. If the first strike doesn't kill, the cavitation bubble finishes the job. And if that doesn't work, the mantis shrimp hits again. And again. Relentless. They've been observed hitting prey items 10 to 20 times in rapid succession. Each strike lands with precision. Same spot, maximum damage. The prey's defenses are irrelevant. The shell, evolved over millions of years to resist crushing, fails. The mantis shrimp eats. But here's the question, how does the club survive? Think about punching a wall 2,000 times at bullet speed. Your hand would shatter, bones would splinter, soft tissue would pulverize. The mantis shrimp's club stays intact. Strike after strike, day after day, year after year. University of California material scientist David Kisailas investigated. Published in Science on February 6, 2025. The club has a multi-layered structure, 3 distinct layers, outer layer, middle layer, inner layer. Each serves a specific function. The outer layer is the impact zone, dense matrix of hydroxyapatite, a mineral, formed into nanocrystal structure. When the club hits, the hydroxyapatite rotates. The nanocrystal structure fractures at the interfaces, then slowly reforms. At low strain rates, the particles deform like a marshmallow, recover when stress is relieved. At high strain, the particles stiffen, fracture, but the fractures are controlled, microscopic. The overall structure remains intact. The hydroxyapatite crystals are arranged in a herringbone pattern. This arrangement distributes stress across multiple planes, prevents cracks from propagating straight through the material. When a crack starts, it encounters a crystal oriented at a different angle. The crack must change direction. This consumes energy, slows the crack growth. Eventually, the crack stops. The material doesn't catastrophically fail. It develops micro cracks that don't compromise structural integrity. The inner layer has a Bouligand structure, named after French researcher Yves Bouligand, who described it in 1972 in crab exoskeletons. Layers of fibers arranged in a helicoidal pattern, each layer rotated slightly relative to the one below, like a spiral staircase of fibers. This structure absorbs and dissipates shock waves, prevents cracks from propagating. If a crack starts in one layer, the helicoidal arrangement redirects it, stops it from reaching deeper layers. The Bouligand structure is used in modern armor design. Composite materials for military vehicles and body armor incorporate helicoidal fiber arrangements inspired by mantis shrimp clubs and other crustacean exoskeletons. The structure provides maximum impact resistance with minimum weight. Researchers have developed synthetic versions using carbon fiber and ceramic composites. These materials outperform traditional steel armor in specific applications. The mantis shrimp discovered this solution 300 million years ago. Humans are just catching up. The middle layer connects the two. Gradual transition zone ensures the hard outer layer doesn't just shatter off the flexible inner layer. 3 layers working together: impact resistance, shock absorption, structural integrity, and the mantis shrimp molts, sheds its exoskeleton, grows a new one. Every few months, each time it molts, the new club is fresh, undamaged, reset to factory condition. Any accumulated microfractures from thousands of strikes get discarded with the old shell. The new club is ready for another few months of hammering prey. Molting is a complex process. The mantis shrimp secretes enzymes that dissolve the connections between the old exoskeleton and the underlying tissue. The animal absorbs calcium and other minerals from the old shell, prepares to build a new one. When ready, the old shell splits along specific seam lines. The mantis shrimp pulls itself out, leaves the old shell behind, like a ghost. The new exoskeleton is soft initially. The mantis shrimp inflates it by absorbing water. The exoskeleton expands, hardens over several hours as minerals are deposited. During this vulnerable period, the mantis shrimp hides, avoids confrontation. Once the new shell hardens completely, the mantis shrimp resumes normal activities. Eyes. The mantis shrimp has the most sophisticated eyes in the animal kingdom. Compound eyes, thousands of tiny elements, each an independent light detector. Set on stalks, operate independently, give unusually broad visual field and keen depth perception. Humans have 3 color-receptive cones: red, green, blue. Mantis shrimps have 12 to 16. They see wavelengths from ultraviolet through visible spectrum into parts we can't perceive. They also see polarized light waves radiating in different directions, useful for detecting transparent prey, communicating with other mantis shrimp, navigating complex reef environments. The 12 to 16 types of photoreceptors don't provide better color discrimination in the way you'd expect. Recent research suggests mantis shrimp process color differently than vertebrates. Instead of comparing signals from different cone types to determine color, mantis shrimp might recognize colors directly based on which photoreceptor is activated. This provides faster color recognition at the cost of some precision. Useful for an animal that needs to make split-second hunting decisions. See prey, identify, strike, all in milliseconds. The visual system is optimized for speed. Cancer detection. The mantis shrimp's sensitivity to polarized light is now informing development of cancer-detecting cameras. Cancerous cells scatter light waves differently than healthy cells due to chaotic structures. Under polarized light, cancer stands out. Medical imaging technology inspired by a 4-inch shrimp that punches crabs. The cameras being developed can detect early-stage cancers that are invisible to standard imaging techniques. Tumors as small as a few millimeters. Before they metastasize, before they become life-threatening. Early detection dramatically improves survival rates for most cancers. A diagnostic tool inspired by mantis shrimp eyes could save millions of lives annually. Clinical trials are underway. Results are promising. Aggression. Mantis shrimp are territorial. They live in burrows, crevices in coral, holes in sandy bottoms. If another mantis shrimp invades, they fight, strike each other with clubs. Recent study published May 9, 2024, in Journal of Experimental Biology by Patrick Green. He introduced pairs of mantis shrimp. Quote, they almost immediately started hitting each other, close, quote, filmed at 30,000 to 40,000 frames per second, 1,000 times faster than regular camera, but mantis shrimp have a defense. They coil their tails in front of their bodies, use the tail like a shield. The tail plates absorb about 20% more of the shock than their body armor alone. Think about a boxer moving with a punch, rolling with the impact. Mantis shrimp do the same. Position their tail, angle their body, reduce damage. Still, the fights are brutal. Both combatants land multiple strikes. The loser retreats. The winner keeps the territory. And both survive because their armor is engineered to withstand the punishment. Exoskeleton tough enough to handle impacts from the fastest punch in the ocean. The fights follow ritualized patterns. Display phase. Both shrimp spread their clubs, show their colors, size each other up. If neither backs down, escalation. They exchange strikes, hit each other's clubs, tail plates, occasionally the carapace. The strikes are controlled, not full force. Testing. If one shrimp lands a solid hit, the other may retreat. If both continue, the fight intensifies. Full force strikes. One shrimp may flip the other, strike the vulnerable underside. The fight ends when one shrimp retreats into a burrow or swims away. Lethal outcomes are rare in laboratory settings, in the wild unknown, but the armor suggests they're adapted to survive intraspecific combat. Aquarium warning. Mantis shrimp in captivity require special enclosures. They can shatter glass, 1/4-inch thick glass, broken. One famous case, a mantis shrimp named Tyson, 4 inches long. April 1998. Smashed through his 1/4-inch thick glass wall. Subdued by nervous attendants. Moved to a more secure facility in Great Yarmouth, not a joke, real incident. Shed Aquarium in Chicago. Their peacock mantis shrimp lives in acrylic, not glass. 3 to 4 times thicker than standard exhibits. Shock absorbent. Even then, aquarium staff stay cautious. The mantis shrimp can punch through acrylic if the walls are too thin. Public aquariums that display mantis shrimp use specially designed tanks, thick acrylic walls, rounded corners to distribute stress, no sharp angles where cracks can start. The tanks are expensive, but necessary. Mantis shrimp are popular exhibits. Visitors love watching them. The bright colors, the alien appearance, The knowledge that this small animal can punch through glass. It's compelling, educational, and slightly terrifying. Species diversity. Over 400 species of mantis shrimp, spearers, smashers. Undifferentiated. Spearers have long barbed appendages, stab soft-bellied prey like fish. Smashers have clubs, batter shellfish and crustaceans. Undifferentiated have appendages between the two forms. All use power amplification, all strike fast. But only smashers and undifferentiated produce cavitation bubbles. Evolution. Stomatopods, the order containing mantis shrimp. Ancient animals. Their evolution predates dinosaurs by around 170 million years. Fossil record shows stomatopods in the Carboniferous. 300 million years ago, they survived 5 mass extinctions. They're still here, still punching. The fossil record includes well-preserved stomatopod specimens showing raptorial appendages similar to modern species. The basic body plan hasn't changed much. This suggests the design is highly optimized. Evolution refined it over hundreds of millions of years, fine-tuned the spring mechanism, perfected the club structure, enhanced the visual system. The modern mantis shrimp is the result of 300 million years of continuous improvement, an evolutionary masterpiece. And here's the cold truth. The mantis shrimp uses the fastest punch in the ocean not to hunt humans, not to conquer territory, just to crack open crabs, to smash snails, to defend a hole in the reef. 4 inches of crustacean delivering more force per body weight than any animal its size. An evolutionary marvel that makes human engineering look clumsy. A living weapon perfected over hundreds of millions of years. And it's swimming in tropical reefs right now, waiting, watching, ready to strike at 50 miles per hour. Number 5. The mimic octopus. The only animal that can impersonate multiple species. Indonesian waters. Sulawesi. Bocas del Toro, Panama. Shallow, muddy river mouths. Sandy estuaries. 1998. Scientists diving, filming, observing, and they see something impossible. An octopus, but not behaving like an octopus. Swimming like a flounder. Striped like a sea snake. Shaped like a lionfish. Changing, adapting, impersonating. Thormoctopus mimicus. The mimic octopus. First discovered off the coast of Sulawesi, Indonesia in 1998, on the bottom of a muddy river mouth, described by Mark Norman, Julian Finn, and Tom Tregenza. Published in Proceedings of the Royal Society B in 2001, Dynamic Mimicry in an Indo-Malayan Octopus. Most animals that mimic can mimic one species, one model, one disguise. The mimic octopus can mimic up to 18 different marine animals. 18. Verified, documented. Not just color change, shape change, behavior change, movement change. The octopus studies its environment, observes the animals around it, then becomes them. Size. About 60 centimeters total length, 2 feet including arms. Diameter approximately that of a pencil at widest point. Small horns protrude from each eye. Natural color is light brown beige, but it usually takes on a more noticeable hue, striped white and brown, to scare off predators by imitating poisonous species. Habitat. Unlike most octopuses that prefer reefs with crevices and holes to hide in, the mimic octopus chooses open sandy areas. River mouths, estuaries, muddy bottoms, exposed, vulnerable, no structures to hide behind. So it uses a different strategy. It becomes something else. The transformations, let's count them. Lionfish. The octopus spreads its arms radially, holds them out like the fish's spines, propels itself through the water column. Brown and white striped pattern appears on the skin. Chromatophores fire. Color cells change. The octopus looks like a venomous lionfish. Predators avoid lionfish. Sharp spines, potent venom. The mimicry works. Lionfish, family Scorpionidae, are apex predators in their native Indo-Pacific range. They hunt small fish and crustaceans. Their spines contain venom that causes extreme pain, swelling, and temporary paralysis in humans. For fish and other potential predators, The venom is often lethal. Lionfish advertise their toxicity with bold stripes and slow, deliberate movements. They're confident. They know nothing will attack them. The mimic octopus exploits this. By looking like a lionfish, it gains the same protection. Predators see the stripes, recognize the threat, avoid contact. Flatfish, zebra sole. Bothus lunatus in Caribbean waters. The octopus pulls all 8 arms flush against its body, flattens out, swims using its siphon to glide over the ocean floor. Looks like a swimming sole. This flatfish is poisonous. Over a 5-day study period, nearly 500 instances of flatfish mimicry were observed by a single individual. Published in Biological Journal of the Linnean Society, 2008, by Roger Hanlon, Lydia Conroy, and John Forsyth. This may be the octopus's preferred disguise. The zebra sole, Zebrius quagga in Indo-Pacific waters, produces toxic mucus in its skin. Fish that bite a zebra sole immediately release it and avoid flatfish thereafter. The octopus mimics both the appearance and the swimming behavior. Undulating movements, close to the bottom, eyes on the upper surface. The octopus even modifies its body outline to match the oval shape of the flatfish. This is the most frequently observed mimicry in wild populations, suggests it's highly effective. Sea snake, banded sea snake, Laticorda species. The octopus hides 6 of its arms in a burrow or crevice, extends the remaining 2 arms parallel to each other, applies black and white bands, moves the arms in an undulating snake-like motion, even adds black dots at the tips to mimic the snake's head pattern. This is the most elaborate impersonation. The octopus only does this when confronted by damselfish, territorial species, known enemies of sea snakes. The octopus detects the threat, chooses the appropriate response, becomes the predator that the damselfish fears. Banded sea snakes are highly venomous, more toxic than most terrestrial snakes. They hunt fish and eels in crevices in coral reefs. Damselfish are small territorial fish that defend their algae farms from herbivores, but damselfish have learned to avoid sea snakes. The snakes are fast, venomous, deadly. When a mimic octopus encounters damselfish, it transforms into the one animal the damselfish cannot afford to ignore. The damselfish retreat. The octopus is safe. Jellyfish. The octopus inflates its mantle, trails its arms behind, drifts like a jellyfish, pulsing, floating. Predators see a jellyfish. Stinging tentacles, not worth the risk. The octopus swims through open, water unharmed. The jellyfish mimicry is less well documented. Observed in aquarium settings and a few wild encounters, the octopus expands its mantle by filling it with water, contracts rhythmically to create pulsing movements. The arms trail behind, resembling jellyfish tentacles. Some scientists question whether this is true mimicry or just a general defensive posture that happens to resemble a jellyfish. More research needed. Small sponge, tube worm, colonial tunicate, sessile animals. The octopus bunches up, stays still, mimics texture and color, blends into the environment as a stationary organism. Predators overlook it. Just another sponge, just another part of the reef. This form of mimicry is called masquerading. The animal resembles an inanimate object or a sessile organism. By remaining motionless, the octopus avoids attracting attention. Predators scan for movement. The octopus doesn't move. The predator's visual system doesn't register it as prey. The octopus waits until the threat passes, then resumes activity. Divers report more. Anemones, mantis shrimp, feather stars, brittle stars, giant crabs, seahorses, crocodile snake eels, stingrays, nudibranchs. These imitations are not yet scientifically verified, but the potential is astounding. 18 species confirmed, possibly more. The decision-making. This is not random mimicry. The octopus appears to choose which animal to impersonate based on the predator it's facing. Example from the 2001 study. When confronted by damselfish, the octopus mimics a sea snake. Damselfish are territorial but afraid of sea snakes. The octopus exploits that fear. When faced with a different predator, it might choose a different disguise. This suggests cognition, decision-making, awareness of the environment and the threats within it. The cognitive abilities required for this behavior are extraordinary. The octopus must recognize different predator species, understand which predator fears which model species, execute the appropriate mimicry. This implies a level of abstract thinking. The octopus isn't just reacting instinctively, it's assessing the situation, choosing a strategy, implementing it. This is problem-solving, planning, theory of mind, recognizing that other animals have knowledge and fears that can be exploited. The brain. Octopuses in general are highly intelligent. Brains contain roughly 300 million neurons. Arms contain roughly 50 million neurons each. Total 400 million neurons distributed through the body. Each arm has its own neural network, semi-autonomous, can make decisions independent of the central brain. Touch, taste, movement, problem-solving. The octopus brain is organized differently than vertebrate brains. There is no cerebral cortex. Instead, intelligence emerges from distributed processing across multiple lobes. The optic lobes process visual information, massive structures, contain more neurons than the central brain. The brachial ganglia in each arm control local reflexes and movement. The central brain integrates information and makes high-level decisions. The system is parallel, multiple processes happening simultaneously. This architecture is efficient for an animal with 8 independent appendages and complex sensory needs. Intelligence examples: opening jars by watching humans, using coconut shells as mobile armor, turning off aquarium lights to avoid excessive brightness, creating mental images to navigate and hunt, learning by observation. These are verified behaviors documented in octopus research. The mimic octopus takes it further. It observes venomous animals, learns their appearance and behavior, replicates them. The coconut shell behavior is particularly striking. Octopuses have been filmed carrying coconut shell halves across the ocean floor. When threatened, they assemble the 2 halves around themselves, creating a protective sphere. This is tool use, planning ahead, carrying a tool for future use. Only a few animal groups demonstrate this ability: great apes, cetaceans, corvids, and cephalopods. The octopus brain, so different from our own, independently evolved complex cognition. Chromatophores, color, changing cells in the skin, thousands of them, each controlled by muscles. Each can expand or contract in milliseconds. When expanded, the chromatophore displays color. When contracted, it becomes transparent. The underlying skin color shows through. By controlling thousands of chromatophores simultaneously, the octopus creates complex patterns: stripes, spots, bands, solid colors, gradients, all in under a second. The chromatophores are arranged in layers. The top layer contains pigments: red, yellow, orange, brown, and black. Below that, iridophores, cells that reflect light, create iridescent blues and greens. Deeper still, leucophores, white cells that scatter light. By coordinating all 3 layers, the octopus can produce virtually any color and pattern. The control is precise. Individual chromatophores can be activated independently. The octopus can create a striped pattern on one arm while keeping the rest of the body plain, or display multiple patterns simultaneously on different body regions: skin texture, papillae, small bumps and ridges on the skin. The octopus can raise or flatten them: smooth skin for gliding like a flatfish, bumpy skin for mimicking a rough sponge, textured skin for blending with coral. The skin itself is a programmable surface. The papillae are controlled by muscles attached to dermal structures. Contract the muscles, the papillae rise. Relax, they flatten. Some papillae are simple bumps. Others are complex branching structures. The largest papillae, found above the eyes, can extend several centimeters. The octopus uses them to break up its outline, disrupt the smooth shape that predators recognize as prey. Combined with color change, texture modification provides 3-dimensional camouflage, movement, behavior mimicry, not just looking like a lionfish, moving like a lionfish, swimming speed, posture, stop-and-go patterns. The octopus watches, learns, replicates. Same for flounders, same for sea snakes. Dynamic mimicry, published in the original 2001 paper. The octopus adjusts its mimicry in real time based on what's happening around it. Lifespan, short, like most shallow water octopuses. Estimated 9 to 12 months based on comparisons with similar species. Macrotritopus defilippi, Atlantic long-arm octopus, lives fast, reproduces once, dies shortly after brooding eggs. The mimic octopus likely follows the same pattern. Reproduction is terminal for most octopus species. Males die shortly after mating. Females brood their eggs, stop feeding, protect the eggs from predators, oxygenate them by blowing water over them. Once the eggs hatch, the female dies. Simulparous reproduction, one reproductive event, then death. This strategy maximizes offspring production, but eliminates the possibility of iterative reproduction. The octopus invests everything in a single clutch. Diet, carnivore, feeds on small fish and crustaceans, echinoderms, uses a jet of water through its siphon to glide over sand while searching for prey, uses slender arms to reach into crevices in coral and holes in the sand, suction cups grab prey, sometimes uses aggressive mimicry, approaches wary prey by mimicking something harmless, like mimicking a crab as a potential mate, gets close then strikes. Predatory deception. Aggressive mimicry is rare in the animal kingdom. It requires the predator to suppress its natural appearance and behavior, imitate something attractive or non-threatening, lure prey close enough to attack. The mimic octopus may use this strategy. Mimic a female crab. Male crabs approach for mating. The octopus captures them. This behavior hasn't been directly observed, but is hypothesized based on stomach content analysis, showing high proportions of crabs in mimic octopus diets. Predators, fish, sea turtles, sea anemones, other larger octopuses. The mimic octopus is small, vulnerable. The mimicry is its primary defense. Without it, life expectancy drops dramatically. The main predators are likely bottom-dwelling fish, groupers, snappers, large wrasses. These fish hunt by sight. They patrol sandy areas looking for prey. The mimic octopus's habitat provides no cover, no reef structures to hide in. The only defense is mimicry. By appearing to be a venomous or unpalatable animal, the octopus discourages attacks. But if the mimicry fails, the octopus can jet away using its siphon, release ink to create a smokescreen, or hide by burying itself in sand. Multiple defensive strategies, all necessary for survival in exposed habitat. Discovery expanded. After the initial 1998 discovery in Indonesia, more sightings followed. Panama, Japan. Great Barrier Reef, Australia, documented 2013 by Coca, published in Marine Biodiversity Records. Philippines, Malaysia. The mimic octopus has a wider range than initially thought, still rare, still difficult to find, but present across Indo-Pacific. Another species joins, Caribbean, Macrotritopus defilippi, Atlantic sand-dwelling octopus, filmed in 5 Caribbean locations, mimicking the swimming behavior and coloration of the common sand-dwelling flounder, Bothus lunatus. Published 2010 in Biological Bulletin by Roger Hanlon and team. First documentation of flounder mimicry by an Atlantic octopus. 4th convincing case of mimicry for cephalopods. The mimic octopus is not alone. Other octopuses are learning the same trick. Here's the twist. Mimicry is considered an evolved behavior, not instinct. Learning. Octopuses observe their environment, process what they see, make decisions based on experience. Some scientists believe the mimic octopus might have some form of consciousness compared to other animals. Self-awareness, cognitive empathy, perspective-taking, imagining itself in the place of the predator, choosing the disguise that will work best. The cognitive semiotic perspective. Study published 2019 by Gomez Moreno, analyzing the sea snake impersonation. The mimicry shows goal-orientedness. The octopus only takes the sea snake form when encountering damselfish, not other animals. This selectivity implies understanding. The octopus knows which predator it's facing, knows which disguise will deter that specific predator, then executes the disguise with precision. This is unparalleled. No other animal switches between multiple models fluidly. Butterflies mimic one toxic species. Flies mimic one bee species. But the mimic octopus impersonates 18, chooses which one, adapts on the fly. This is the evolutionary perfect storm: flexible, soft body, skin capable of instant color and texture change, brain sophisticated enough to observe, learn, and replicate, environment filled with venomous species to copy, predators everywhere creating selective pressure, all conditions met. Result: the ultimate master of disguise. And here's the cold reveal. We only discovered this animal 27 years ago, 1998, less than 3 decades. It's been living in those river mouths for millions of years, mimicking, adapting, surviving. We just weren't looking. How many other species in the ocean have abilities we haven't documented yet? How much more is hiding in the mud and sand, waiting for a researcher with a camera to finally see it? The ocean is vast. The mimic octopus proves that even shallow water near shore holds creatures that rewrite what we think is possible. Swimming, watching, changing, becoming, whatever it needs to be to survive another day. Number 6. The vampire squid. The living fossil from the oxygen minimum zone, deep ocean, 200 to 3,000 feet, 600 to 900 meters, permanent darkness, crushing pressure. Oxygen levels so low most animals suffocate. And there, drifting, a squid. Except it's not a squid, not an octopus either. It's something older, something that split from both lineages over 300 million years ago. Vampiroteuthis infernalis, vampire squid from hell. The name alone tells you this is not a normal animal. Discovered in 1883 by German marine biologist Carl Chun during the Valdivia deep sea expedition. But scientists didn't understand it. Looked like a squid. 8 arms like an octopus, soft, gelatinous body, small size, maximum 30 centimeters total length, 12 inches, mantle length about 15 centimeters, 6 inches, weighs almost nothing, less than half a kilogram, color, deep red to black depending on lighting. In darkness it appears black. When illuminated it shows dark red or purple hues. Large blue eyes, huge relative to body size, 3 centimeters diameter on a 15 centimeter body, needs big eyes to capture any available light in the deep. The arms, 8, but they're connected by a web of skin, a cloak. When threatened, the vampire squid pulls all its arms above its body, inverts the web, creates a defensive posture that covers the entire animal. The underside of the web is lined with fleshy spines called cirri, rows of them. When the cloak is inverted, the cirri face outward, creates a spiky ball. Predators see something unappetizing, difficult to swallow. The vampire squid holds this position, drifts, waits for the threat to pass. But that's not the strangest defense. The vampire squid has bioluminescent photophores all over its body, tips of the arms, underside of the cloak. When threatened, it ejects glowing mucus from the tips of its arms, a cloud of bioluminescent particles, confuses predators. The vampire squid then jets away in a different direction. The predator attacks the glowing cloud. The squid escapes. The bioluminescent mucus is unique. Unlike ink from shallow water squid, which creates a dark cloud, the vampire squid's defense glows. The photophores in the ejected mucus remain active for several minutes, producing pinpoints of blue-green light. From the predator's perspective, the cloud looks like a swarm of bioluminescent prey. The predator investigates or attacks the cloud. By the time it realizes the deception, the vampire squid is long gone, and it can turn itself inside out, literally, pulls its mantle over its head, exposes the darker underside, changes its visible profile, another defensive trick. Few animals can invert their entire body. The vampire squid does it routinely. Movement, not a strong swimmer. Slow, drifts using its single pair of fins. Fins look like large ears on the sides of the mantle. Flaps them slowly. Generates gentle propulsion. Can also use jet propulsion. Expels water through its siphon. But the vampire squid doesn't. Jet often. Oxygen is scarce in the oxygen minimum zone. Vigorous activity burns oxygen. The vampire squid conserves energy, moves slowly, efficiently. The oxygen minimum zone is one of the most challenging environments on Earth. Oxygen concentration drops below 0.5 milliliters per liter. Most marine animals require at least 2 to 3 milliliters per liter. Below that, they suffocate. Their metabolic processes shut down. But the vampire squid has adaptations. Extremely low metabolic rate. Uses oxygen at a fraction of the rate of other cephalopods. Blue blood, hemocyanin, copper-based oxygen transport molecule, more efficient than hemoglobin in low oxygen conditions. Large gills relative to body size. Maximizes oxygen extraction from the water. Diet. This is where the vampire squid breaks the mold. Most cephalopods are active predators. Hunt live prey. The vampire squid doesn't hunt. It's a detritivore. Eats marine snow. Dead organic matter drifting down from the surface. Bits of dead plankton. Fecal pellets. Molted exoskeletons. Disintegrating tissue. This debris rains down constantly in the deep ocean. The vampire squid intercepts it. Marine snow is the primary source of nutrition for deep-sea ecosystems. Surface waters are productive. Phytoplankton blooms. Zooplankton graze. Fish feed. All this activity produces waste. Dead organisms, fecal material, it sinks, forms aggregates, flakes that drift downward like snow. By the time it reaches 600 to 900 meters, it's been picked over by countless organisms. The nutritional value is low, but it's constant, always falling. The vampire squid evolved to exploit this resource that other cephalopods ignore. The feeding mechanism. 2 filaments, long, thin, retractable tentacles, different from the 8 arms. These filaments extend up to 8 times the mantle length, covered in sticky mucus. The vampire squid extends the filaments into the water, lets them drift, captures marine snow particles on the sticky surface, then retracts the filaments, runs them through its arms, scrapes off the collected debris, packages it into a mucus ball, eats it. This feeding strategy is unique among cephalopods. Published 2012 in Proceedings of the Royal Society B. Vampire squid feed on detritus, not live prey. Energy efficient. Doesn't require chasing anything. Doesn't require overpowering struggling animals. Just collect the falling debris. The oxygen minimum zone is too low in oxygen for active hunting. The vampire squid adapted to a completely different niche. The filaments are retracted into pockets between the arms when not in use. When extended, they can reach over a meter in length, thin as threads, nearly invisible. The vampire squid extends one or both filaments depending on current and food concentration. The mucus coating the filaments is continuously secreted. Particles stick immediately. The filament loading process can take hours. The vampire squid is patient, drifting, collecting. Eventually, the filament is retracted. The accumulated marine snow is consumed. The process begins again. Reproduction. Little is known. Scientists have collected very few specimens. Observations in the wild are rare. What we know comes from preserved specimens and limited ROV footage. The female lays large eggs, huge compared to body size. Each egg is about half a centimeter, dozens of them. She releases them into the water. They're neutrally buoyant, float at the same depth, hatch into juveniles that look like miniature adults. No planktonic larval stage. Direct development. The large egg size indicates high parental investment. Each egg contains substantial yolk, provides nutrition for extended development. The juvenile that hatches is well developed, ready to begin feeding immediately. This strategy is common in deep-sea animals, producing a few large, well-provisioned offspring rather than many small, vulnerable larvae. In the deep sea, food is scarce. Larvae would have difficulty finding enough to eat. Better to develop inside the egg, emerge ready to exploit the adult food source. Lifespan. Estimated several years, maybe longer. The vampire squid grows slowly, matures slowly, reproduces multiple times over its life. This is unusual for cephalopods. Most cephalopods reproduce once and die, semiparity, but the vampire squid might be iteroporous, multiple reproductive cycles. The low oxygen environment slows metabolism, extends lifespan. Slow metabolism means slow aging. Cells divide less frequently. DNA accumulates damage more slowly. Telomeres shorten at reduced rates. The vampire squid might live a decade or more, producing eggs multiple times, spreading reproductive effort across years. This increases the probability that at least some offspring survive. In unpredictable environments, iteroparity is advantageous. If conditions are bad one year, there's always next year. The evolutionary position. Vampire squid belong to the order Vampiromorphidae. They are not squids, not octopuses. They are the only surviving members of their order. Sister group to both squid and octopuses. Split from common ancestor over 300 million years ago. Living fossil. The vampire squid represents an ancient lineage, survived mass extinctions, adapted to one of the most extreme environments in the ocean. Anatomy is intermediate, 8 arms like octopus, but has fins and a gladius, an internal shell like squid. The gladius is reduced, vestigial, soft and flexible, not rigid. Intermediate characteristics. Bridge between octopus and squid. Phylogenetically distinct. The fossil record shows vampiromorphids were more diverse in the past. Multiple species, varying sizes and body plans, but only Vampiroteuthis infernalis survived to modern times. The others went extinct, possibly due to changing ocean conditions, expanding oxygen minimum zones during some periods, contracting during others. The surviving species is the one best adapted to the most extreme low oxygen conditions. It persisted. The others did not. Oxygen minimum zone. Layer in the ocean where oxygen concentration drops to less than 0.5 milliliters per liter. Most marine animals can't survive. Fish suffocate, crustaceans die. But the vampire squid thrives. It has the lowest metabolic rate of any cephalopod, uses oxygen so efficiently it can live where others can't. Blue blood, hemocyanin, copper-based oxygen transport, more efficient than hemoglobin in low oxygen conditions. Large gills relative to body size, extracts every molecule of oxygen from the water. The oxygen minimum zone forms due to microbial activity. Bacteria consume oxygen while decomposing sinking organic matter. In regions where decomposition rates are high and water circulation is limited, oxygen levels plummet. The zone typically occurs between 200 and 1,000 meters depth, varies by location. The eastern Pacific off Mexico and Peru has extensive oxygen minimum zones, the Arabian Sea, parts of the Atlantic. These zones are expanding due to climate change. Warmer water holds less dissolved oxygen. Ocean circulation is changing. The zones are growing thicker, moving into shallower depths. This benefits the vampire squid. More habitat, but it squeezes other species, compresses their vertical range, forces them into narrower bands of livable water. The vampire squid is one of the few winners in a warming ocean. Predators, few. The deep ocean has few predators in general. Large fish, deep-diving marine mammals, sperm whales, pilot whales, elephant seals, all dive to the oxygen minimum zone, all hunt there. The vampire squid's defenses work. Bioluminescent mucus, cloak inversion, cirri spines, enough to deter most attacks. And if a predator does grab it, the vampire squid can autotomize, drop an arm, sacrifice it, escape. The arm will slowly regenerate over months. Sperm whales are probably the main predators. They dive to over 2,000 meters, hold their breath for 90 minutes, hunt squid in the deep. Vampire squid are small, not preferred prey, but opportunistically taken. Beaks of vampire squid have been found in sperm whale stomachs, not many. The vampire squid's defensive strategies are effective. Most escape. Discovery history. First collected 1883 by HMS Challenger expedition. Preserved specimen. No living observations for over a century. Not until deep-sea submersibles and ROVs became available in late 20th century did scientists see vampire squid alive. Monterey Bay Aquarium Research Institute, MBARI, multiple ROV dives between 2000 and 2010, hundreds of hours of footage. Vampire squid behavior documented, feeding strategy revealed, defensive postures recorded, published 2012. The MBARI observations revolutionized understanding. Before the ROV footage, scientists assumed vampire squid were active predators like other cephalopods. The presence of a beak suggested carnivory. The arms and filaments were misunderstood. But video showed the truth. The vampire squid drifts, extends filaments, collects marine snow. A completely unexpected feeding strategy. The paper detailing these findings generated widespread media coverage. The vampire squid became famous, not as a fearsome predator, but as a gentle detritivore quietly drifting in the deep. Cleaning up the ocean's garbage, a very different image than the hellish vampire the name suggests. And here's the reframe. The vampire squid is not a vampire. It doesn't suck blood, doesn't hunt living prey. The name is a misnomer, based on appearance. Red cloak, large eyes, infernal habitat. But the animal is actually gentle, feeds on debris, drifts quietly, avoids confrontation, lives in one of the harshest zones on the planet using strategies no other cephalopod employs. 300 million years old as a lineage, survived the Permian extinction, the Cretaceous extinction, the ice ages, the warming periods. It's still here, still drifting, still collecting marine snow, a living window into deep evolutionary history, the last of its kind, and almost nobody knows it exists because it lives where humans can barely reach, in permanent night, in suffocating water, in the vampire squid's hell, which for the vampire squid is home. Number 7. The barrel-eye fish. The fish with a transparent head and telescopic eyes. Imagine looking up, through your skull, seeing the sky above you, not through your eyes in their normal position, through eyes mounted inside your transparent skull pointing straight up. That's the barrel-eye fish. Macropinna, microstoma. And for 70 years scientists thought they understood it, until ROV footage in 2004 proved everyone wrong. The fish, small, 15 centimeters long, 6 inches, lives in the deep sea, 600 to 800 meters, 2,000 to 2,600 feet, twilight zone. Some light filters down from the surface, but not much. Mostly darkness, the head, the skull, transparent, clear, see-through, filled with fluid, and inside. 2 cylindrical eyes, tubular, mounted vertically, pointing straight up, covered by a green lens that filters out sunlight, enhances contrast in the dim light of the deep. The original understanding. Specimens dredged up from the deep arrived mangled. The transparent dome over the head was always destroyed by the nets, torn apart by pressure change. Scientists only saw dead fish with damaged heads. They described the eyes as fixed, pointing upward, rigid. The fish looks up, sees prey silhouetted against the faint light from above, swims up, opens its tiny mouth, eats zooplankton. Standard explanation, published 1939 by Chapman, accepted for decades. 2004. Monterey Bay Aquarium Research Institute, MBARI, ROV Tiburon. Diving in Monterey Bay. Camera spots a barrel-eye, alive, intact, transparent dome, pristine. And the eyes, they're not fixed, they rotate. Inside the head, the fish looks up, then the eyes swivel forward. The fish looks straight ahead, then the eyes rotate back up. Flexible, mobile, completely unexpected. Bruce Robertson and Kim Reisenbichler from MBARI analyzed the footage, published 2009 in Copea. The transparent head isn't just a shield for the eyes, it's a viewing window. The fish can track bioluminescent prey above, follow it, then rotate its eyes forward to see where it's going as it swims. Solves the problem of tubular eyes. Tubular eyes give great resolution in One direction but narrow field of view. The barreleye gets around this by moving the eyes inside the fluid-filled dome. The discovery answered a long-standing question. How does a fish with upward-pointing eyes catch prey that's above it? The obvious answer was that it swims straight up, but observations showed barreleyes swimming horizontally, eyes pointed up. Made no sense, until researchers realized the eyes could rotate. When the fish detects prey above, it watches, tracks the prey's movement. When ready to attack, the eyes swivel forward, the fish swims horizontally, positions its mouth under the prey, sucks it in. Eyes swivel back up, returns to scanning mode. The green lenses, filters. The barreleye lives in an environment where bioluminescence is common. Many creatures in the twilight zone produce light, blue-green wavelengths mostly. The green filter blocks ambient downwelling sunlight. Blue wavelengths from the surface enhances the bioluminescent signals from prey, makes glowing animals stand out. The fish sees them more clearly through the filter than without it. The physics of this are elegant. Sunlight penetrating from the surface is predominantly blue at these depths. Red and yellow wavelengths are absorbed in the upper water column. By 600 meters, only blue light remains, but bioluminescence produces light at slightly different wavelengths, more green. The green filter reduces the blue sunlight while transmitting the green bioluminescence, increases contrast. The prey glows brighter against a darker background. The barreleye detects prey that other fish without green filters cannot see as clearly. Diet: zooplankton, small jellyfish, siphonophores, drifting invertebrates. The barreleye hovers in the water column, nearly motionless, waits. Scans above with its tubular eyes, detects prey silhouetted against the faint light, rotates eyes forward, swims up carefully, opens its small mouth, engulfs the prey, returns to hovering position. Energy efficient, doesn't chase, just intercepts. Siphonophores are colonial organisms related to jellyfish, long trailing tentacles. Some species grow to 30 meters or more in length. They drift in the water column, catch zooplankton with stinging cells. The barreleye steals prey from siphonophore tentacles, swims along the tentacles, picks off captured prey. The siphonophore can't pursue, it drifts passively. The barreleye takes advantage. This behavior, called kleptoparasitism, provides a steady food source. The siphonophore does the hunting. The barreleye reaps the benefits. The body, dark brown to black, covered in large scales. Fins are relatively large for slow, precise movement. Small mouth, weak jaws, not a predator of large or fast prey. Specialized for small, delicate organisms. The digestive system is simple, suited for soft-bodied zooplankton that don't require much breakdown. The scales are pigmented to match the dark water, reduces visibility from below. Predators looking up see a dark silhouette that blends with the dim background. The fins are transparent or lightly pigmented. When the fish is motionless, it's nearly invisible. Only the eyes, hidden inside the transparent dome, might give it away. But the dome refracts light, distorts the outline, makes the fish harder to recognize as a fish. Visual camouflage through transparency and shape distortion. Depth range. Most commonly found between 600 and 800 meters, but individuals have been recorded shallower and deeper. Follows prey distribution, follows food. The twilight zone is a productive layer. Dead and dying plankton sink from surface. Deeper animals migrate up at night to feed. The barreleye positions itself in this zone, intercepts food as it passes. The twilight zone, also called the mesopelagic, is a transition layer. Above, the epipelagic, sunlit surface waters. Below, the bathypelagic, perpetual darkness. The mesopelagic receives faint sunlight, enough for vision, not enough for photosynthesis. Animals here have large eyes, bioluminescence, strategies to detect prey and avoid predators in dim light. The barreleye is typical, extreme visual adaptations, behavior optimized for a low-light environment. Discovery, first described 1939, but the transparent dome wasn't understood. Specimens were damaged. Only after live observations in 2004 did the full picture emerge. The head is transparent, the eyes rotate, The fish uses the dome as a protective barrier and a window. All features working together for a survival strategy perfectly suited to the deep twilight zone. The 1939 description was based on a single damaged specimen. The skull was crushed, the eyes visible but the surrounding tissue destroyed. Chapman, the describing scientist, did his best with what he had, but without seeing a live specimen he couldn't know about the transparent dome. Couldn't know the eyes rotated. His description was accurate for what he could observe, but incomplete. This highlights a common problem in deep-sea biology. Many species are known only from damaged specimens. Their true anatomy and behavior remain mysteries until someone observes them alive in their natural habitat. Other barrel-eye species exist. Opisthoproctidae family. About 19 species total. All have upward-pointing tubular eyes. Some have transparent domes. Some don't. Some have eyes that rotate. Some have fixed eyes. But Macropina microstoma is the most studied, the clearest example of the transparent skull adaptation. Different species show variations on the theme. Some opisthoproctids have eyes that point obliquely upward and forward. Others have one eye pointing up and the other forward. Permanent asymmetry. The diversity suggests the family is exploring different solutions to the same problem. How to see in the twilight zone, how to detect prey, how to avoid predators. Each species has its own answer. The transparent dome is one solution, not the only one, but perhaps the most elegant. Behavior in captivity. Extremely difficult. Few aquariums have successfully kept barrelies alive. The Monterey Bay Aquarium managed to display individuals temporarily. The fish requires specific water temperature, low light, gentle water flow, no strong currents. The transparent dome is fragile. Collisions with tank walls can damage it. Most specimens die within days. The deep sea fish does not adapt well to captivity, better studied in the wild with ROVs. The pressure change is a major problem. Barrel eyes live at 60 to 80 atmospheres of pressure. Bringing them to surface 1 atmosphere causes gases in their tissues to expand. Swim bladders rupture, internal organs are damaged. Even if captured alive, they rarely survive transport. Those that do arrive at aquariums alive are stressed, injured internally. They refuse food, fade, die. Keeping deep sea fish alive requires specialized equipment, pressure tanks, slowly decompressing the fish over days. Few facilities have this capability. Research continues, but for now, barrelies are best studied where they live. Evolutionary advantage. Why evolve a transparent head? Protection and visibility. The eyes are large, delicate, vulnerable to damage. If they were external, jellyfish tentacles could sting them. Particles could scratch them. The transparent dome shields them from physical contact, and the fluid inside the dome provides cushioning, stabilizes the eyes, allows them to rotate smoothly. The tubular shape gives superior light gathering, like a telescope, concentrates faint light onto the retina, increases sensitivity. The barrel eye can see in conditions where flat eyes would fail, sees prey that other fish can't detect, sees predators approaching from above. The upward gaze is both offensive and defensive. Tubular eyes are found in several deep sea fish families. Hatchetfish, spookfish, all have similar problems. Need to detect faint light. Need high visual acuity. Tubular eyes provide both, but restrict field of view. The barrel eye solution, rotating the eyes inside a transparent dome, is unique. Other families solved the problem differently. Multiple eyes, mirrors instead of lenses, split retinas, convergent evolution toward highlight sensitivity through multiple pathways, predators, few specifics known, larger fish in the twilight zone, deep diving squid, possibly marine mammals. The barrel eye's strategy is avoidance. Stay still, don't create vibrations, don't attract attention. Transparency helps. The body is dark but small. The eyes are hidden inside a transparent dome, harder to see, harder to target. The reframe. For 70 years, scientists described the barrel eye based on dead, damaged specimens, fixed eyes, limited vision, standard deep-sea adaptations. Then, live footage revealed the truth. The eyes rotate. The transparent dome is a functional structure. The fish has a level of visual capability and flexibility that was completely unexpected. It took live observation to correct the record. And here's the final piece. The barreleye is not rare. ROV surveys find them regularly in Monterey Bay and other deep ocean sites. They're just rarely seen because they live in a zone humans don't visit, 600 to 800 meters, too deep for most divers, too shallow for typical deep-sea exploration. The twilight zone, the overlooked zone, full of animals like the barreleye, adapted in ways we barely understand, invisible until an ROV happens to point a camera at the right spot at the right time. The barreleye fish proves that even well-studied animals can hide secrets. Even animals described 80 years ago can surprise us when we finally see them alive. The ocean is not empty. It's not simple. Every depth has creatures engineered by evolution to exploit that specific environment. And some of those creatures, like the barreleye, have solutions so strange that when we finally discover them, we have to rewrite the textbooks. A transparent skull, rotating eyes, a fluid-filled dome, all real, all functional, all swimming in the ocean's twilight zone right now. These are the 7 most amazing ocean creatures you'll ever hear about. The blue whale, 190 tons of pure mass, the largest animal that has ever existed. The colossal squid, eyes the size of dinner plates, first filmed alive 100 years after we named it. The immortal jellyfish, reverses its aging, becomes young again, dodges death indefinitely. The mantis shrimp, punches at 50 miles per hour, club that never breaks, cavitation bubbles that hit like bullets. The mimic octopus, 18 species impersonated, changes shape and behavior on demand, masters of disguise. The vampire squid, 300-million-year-old lineage, eats marine snow in the oxygen minimum zone, living fossil from hell. The barreleye fish, transparent skull, eyes that rotate inside a fluid-filled dome, hidden in the twilight zone for 70 years before we saw the truth. 7 species, 7 impossible abilities, all documented, all verified, all swimming in our oceans right now. Not myths, not legends, not exaggerations, just evolution doing what it does best, creating animals so perfectly adapted to their environments that when we finally discover them, we can barely believe they're real. But here's the final reveal that changes everything. These 7 are just the beginning. The ocean covers 71% of Earth's surface. 95% of it remains unexplored. We've mapped more of Mars than our own ocean floor. And every time we send an ROV down, every time we drop a camera into the deep, every time we study a muddy estuary or a twilight zone, current, we find more. Carnivorous sponges, glass worms that glow, squid with elbows, shrimp that farm bacteria, jellies bigger than blue whales, worms that live on whale bones, fish that walk, octopuses that use tools, crabs that decorate themselves with anemones, sea slugs that steal chloroplasts from algae and photosynthesize like plants, all real, all documented in the last 2 decades. The Schmidt Ocean Institute's research vessel Falkor 2, expeditions in 2024 and 2025, discovered hundreds of new species: death ball sponges, iridescent scale worms, juvenile colossal squid filmed for the first time, first footage of glacial glass squid, methane seep communities in Chilean waters, hydrothermal vent ecosystems, coral gardens over 2,000 feet deep. All new, all documented, all published in journals like Nature and Biodiversity Data Journal and Biological Bulletin. The discoveries don't stop, they accelerate. And most of these creatures live in places we thought were empty. The deep ocean, the twilight zone, oxygen minimum zones, sandy plains, muddy estuaries, the boring places. The places with no structure, no reefs, no obvious life. But when you look closer with the right tools at the right time, life, everywhere, adapted, thriving, surviving in conditions that would kill surface animals in seconds. So when someone tells you the ocean is explored, when someone says we know what's down there, when someone claims we've documented all the large animals, remember these 7. Remember that we filmed a colossal squid alive for the first time this year. 2025. Remember that the barrel-eyes rotating eyes were only discovered 20 years ago. Remember that the mimic octopus was completely unknown until 1998. 27 years ago, an animal that impersonates 18 species, swimming in shallow water, near shore, and we never noticed. The ocean is not empty. It's not understood. It's vast, it's dark, it's hostile, and it's full of animals that evolution spent hundreds of millions of years perfecting. Animals with abilities we're only beginning to document. Animals that punch like bullets. Animals that cheat death. Animals that change shape. Animals heavier than any creature that ever walked on land. All real, all verified, all waiting in the water for the next ROV, the next submersible, the next scientist with a camera and the courage to look deeper. These are the 7 most amazing ocean creatures, but there are thousands more, and we're just starting to find them.